May peace be on all of you. Myself, Dr. Majid Khan, Associate Professor, Loknete Dr. J.D. Pawar, College of Pharmacy, Kalwan, Nasik. I welcome all of you in this lecture 5 of anti-cancer agent. In this video, that is in lecture 5, we will cover anti-cancer antibiotic and antimitotic, that is Vinca alkaloids. After completion of this topic, student able to understand the activation, mechanism of action and metabolism of anthracycline anti-cancer, understand mechanism of action and uses of Vinca alkaloids. So first is anthracycline anti-cancer antibiotic. Anthracycline is characterized by planar oxidized anthracene nucleus fused to cyclohexane ring, which is connected via glycosidic linkage to amino sugar. So it is a general structure of anthracycline derivative. So it contains this three ring is anthracene, which is connected to the fourth ring, that is cyclohexane ring. This cyclohexane ring is connected glycosidically with amino sugar. So it is a amino sugar. So it having three part: anthracene, cyclohexane, then amino sugar. So it is a ring A, this one is a ring B, this one is a ring C, and this one ring is D. So here there are four examples in this anthracycline. First one is daunorubicin, doxorubicin, idarubicin and carminomycin. So here the difference is substituent R1 and R2. In case of daunorubicin, R1 is methoxy group, R2 equal to hydrogen. Doxorubicin, R1 methoxy group, R2 hydroxyl group. Idarubicin, R1 hydrogen, R2 hydroxyl group. Carminomycin, R1 hydroxyl group, R2 hydrogen. So these are the examples of anthracycline derivative. Regarding mechanism of action or regarding the binding of this drug to the DNA, the studies of mechanism by which anthracycline exhibit their cytotoxic effect initially focus on the ability of the compound to associate with DNA resulting from intercalation of their planar ring system supported by binding of amino sugar. Subsequent work focused on the ability of these compounds to generate free radical. Although free radical generation is thought to be important in toxicity, currently the accepted mechanism involve intercalation followed by inhibition of topoisomerase second enzyme, resulting in strained breakage leading to apoptosis. Anthracycline are considered specific for the S phase of cell cycle. So regarding mechanism of action, this drug act through the intercalation mechanism, that it intercalate with DNA molecule. Mainly this ring are intercalated in DNA ring, and there is also generation of free radical, but it is associated with toxicity of this drug. This drug also inhibit topoisomerase second enzyme, which causes breaking of DNA strand, and this drug is S-phase specific drug. Regarding intercalation, the general feature shows ring B and ring C. So it is a ring B and C. B and this one is C. Intercalated between CG base pair. CG base pair means cytosine guanine base pair, which is a important base pair in DNA. So this B and C ring intercalated between CG base pair and ring D intercalated into major groups, and the sugar moiety along with ring A. The sugar moiety along with ring A intercalated in minor groups. So what is major groups and minor groups? So it is a DNA structure, it is a major groups. This gap is major groups, this one is minor groups. So B and C ring of anthracycline intercalated between CG base pair, ring D intercalated in major groups, and ring A along with amino sugar intercalate in minor groups. The amino sugars seem to play an especially important role compound lacking this function fail to inhibit the enzyme. So presence of amino sugar is also important. If the compound not contain this amino sugar, it does not inhibit the topoisomerase second enzyme. The formation of covalent bond between anthracycline and DNA also supported by several studies in which formaldehyde is produced by oxidation of cellular component, or this oxidation result from the production of ROS, reactive oxygen species, such as hydrogen peroxide, that is H2O2, which are generated during redox cycling of anthracycline. The generated formaldehyde may then form methylene bridge between the amino group of anthracycline and two amino group of guanine base in DNA. So here the mechanism of action. So anthracycline first undergo redox cycling reaction to form the hydrogen peroxide. So in next slide we see what is the redox cycling reaction. So here first anthracycline undergo a redox reaction to form the hydrogen peroxide, that is H2O2. Then this H2O2 again form there is a formaldehyde after the reaction with cellular component. So this formaldehyde is formed. Then this formaldehyde react with another molecule of anthracycline. So here is a anthracycline, then it react with the formaldehyde. So this NH2 group of anthracycline, this one is amino sugar NH2 group, react with this formaldehyde by this mechanism. So here it is the mechanism. So here there is a RNH2. RNH2 means this. Suppose we will consider R equal to this whole structure of anthracycline, and this NH2 is equal to this NH2. So here RNH2 means it is a anthracycline NH2, that is NH2 of amino sugar of anthracycline, and this one is formaldehyde which is formed here. So by this mechanism, so here lone pair attack to this carbon, it become O minus, then hydride transfer, it become OH like this. Then there is a removal of water molecule, hydrogen remove here, this bond shifted here, OH minus. So here there is a formation of RN double bond CH2. So here, so if you see here, there is a R means this structure, then N double bond CH2. So anthracycline which contain amino sugar, which contain amine group, react with this formaldehyde by this mechanism, and there is a formation of bridge. So here N double bond CH2 is formed. Then this bridge form the bridge between the amino group of guanine. So it is a DNA part guanine. So as we know, DNA consists of various bases. So it is a guanine base. So after reaction with formaldehyde, the anthracycline react with this NH2 group of guanine. So here again lone pair of NH2 attack to this carbon, then this bond is break, it become N minus, then hydride transfer here. So like this it react. So here is a complex between the anthracycline and DNA bases. So by this way anthracycline inhibit the DNA bases. So again I repeat, it is anthracycline, it undergo redox cycling reaction to form the free radical that is called as a reactive species, that is hydrogen peroxide. Then this hydrogen peroxide, that is a reactive species which interact with the cellular component to form the formaldehyde. Then this formaldehyde again react with the anthracycline. So formaldehyde react with amino group of amino sugar of anthracycline by this mechanism. So here there is a formation of N double bond CH2 group. Then this group react with NH2 group of guanine, which is a DNA part. Then it form this type of complex. So by this way anthracycline react with DNA bases and interrupt the inhibit the DNA synthesis. Although now if we see it, so it is about the mechanism of action. Now the toxicity of anthracycline, that is called as adverse effect. Although the anthracycline produces several adverse effects that are typical for antineoplastics, but the cardiotoxicity is a special concern with this class of agent. The associated cardiopathies and congestive heart failure have been related to the ability of this compound to undergo redox cycling reaction. The C ring of anthracycline is subject to 1 or 2 electron reduction by several different enzymes including NADPH oxyreductase and cytochrome P450 reductase enzyme. So here the main toxicity is cardiotoxicity. So as we discuss in mechanism action, anthracycline undergo redox cycling reaction. So it is a redox cyclic reaction in which C ring. So this one is C ring. So here free radical is formed, that is superoxide radical. So here this double bond shifted here inside the ring, then this pi bond shifted here, and then it become O minus. So like this. So it is a reversible reaction. Again this product is formed, so it is called as cyclic redox reaction. So during this reaction superoxide is produced. So it is a free radical superoxide. So it is called as redox reaction. Again I repeat, C ring involved here. So C ring undergo the 1 or 2 electron reduction. Then this bond shifted here, this bond shifted here, then it become O minus. So the radical is normally converted into hydrogen peroxide. So here the superoxide is produced and this superoxide converted into hydrogen peroxide by superoxide dismutase enzyme. So during this redox reaction, superoxide free radical is produced, which is converted into hydrogen peroxide by the enzyme superoxide dismutase. So here the H2A2 is converted into water and oxygen by the enzyme catalase. However, the production of superoxide is also associated with the release of iron, that is Fe2 plus, from intracellular store, which may be chelated by anthracycline. The iron, that is Fe2 plus, then divert this normal detoxification pathway by catalase so that more potent radical, hydroxyl radical, are produced. So here during redox reaction superoxide is produced, which is converted into hydrogen peroxide by the enzyme superoxide dismutase. Free radical also causes release of Fe2 plus from the storage site, and this Fe2 plus convert this H2O2 into hydroxyl free radical. So this Fe2 release divert this normal reaction to this harmful reaction. So it is a normal process which is harmless. H2A2 conversion into water and oxygen, it is a harmless, and enzyme is catalase. But due to release of this Fe2 plus, this process is diverted to harmful reaction, that is conversion of H2O2 into hydroxyl free radical. So Fe2 plus is responsible for formation of free radical, which is toxic to our body. So Fe2, in presence of Fe2, this H2O2 converted into free radical instead of water and oxygen. Myocardial cell possess lower level of catalase and therefore are less able to detoxify the hydrogen peroxide that result from redox cycling. This resulting in increased level of free hydroxyl free radical, for which detoxifying pathway does not exist, and therefore cellular damage occur. The true hydroxyl free radical causes single strand break in DNA, which activate P53 and enhance apoptosis in cardiac cell. So here H2O2 converted into hydroxy free radical. The cardiac cell having less amount of catalase, therefore this free radical is formed, and this free radical damage the cardiac cell and produce the cardiac toxicity. Studies have shown that doxorubicin administration stimulate both intrinsic and extrinsic pathway of apoptosis, and this has been linked to increase the production of H2O2 and hydroxyl free radical. So cardiotoxicity is due to formation of this free radical, and this free radical is formed when there is a release of this Fe2 plus, which convert this H2O2 into the free radical and which causes cardiac toxicity. So it is about the adverse effect of anthracycline, that is cardiac toxicity. So in summary, anthracycline undergo redox reaction to form the superoxide free radical. Then the superoxide converted into H2O2 by the enzyme superoxide dismutase. Normally this H2O2 is converted into harmless product water and oxygen by the enzyme catalase. But in presence of Fe2, which is released during this reaction from the storage site, and this Fe2 plus convert this H2O2 into the harmful product, that is hydroxyl free radical. So in short, this Fe2 plus divert this normal reaction into the harmful reaction. So in cardiac cell there is a less level of catalase, therefore this free radical is formed which causes the damage to the cardiac cell. So it is the cardiotoxicity of anthracycline. Now, strategy used to reduce this cardiac toxicity. So several strategies have been developed to reduce the cardiac toxicity, and that first one is slow infusion over 48 to 96 hours versus bolus given over 15 minutes. So it means slow infusion means anthracycline slowly administered over the 48 to 96 hours. So anthracycline solution form slowly injected, that is called as infusion, so that there is a less chances of free radical formation, followed by bolus. Bolus means a rapid administration over the 15 minutes. So there is a less chances of formation of this free radical. So it is the first strategy, which is an older strategy to reduce the cardiotoxicity side effect of anthracycline. Second, that is use of antioxidant like vitamin A, C and E. So antioxidant neutralize the free radical, that is oxidant. Third, by using thiol containing reducing agent such as acetylcysteine. So this reducing agent reduce the Fe2 plus into Fe3 plus, therefore prevent the formation of free radical. Then the fourth method, which is the important one, alternative method used to chelate the iron required for activation of H2O2, and for this purpose dexroxazone is mainly used. So here the antidote is dexrazoxane. It is a dexazoxane or ADTA to chelate the iron. So as we seen in toxicity, iron play an important role in formation of free radicals. So this drug, that is called as doxazoxane, is used. This doxazoxane is rapidly taken up into the myocardial cell, where it is subsequently hydrolyzed by two-step process to yield diamide acid, which chelate the free iron, and then iron bound to anthracycline and is similar chelating agent is ADTA. So it is a antidote doxazoxane. So it is a structure of doxazoxane. First it is hydrolyzed into the diamide acid. So here water molecule is added, this bond is break. So hydrogen is added here, hydroxyl group added here, like this. Similarly here. So it gives two-step process. So it is the activation of this antidote. So it is converted into diamide. Then this diamide chelate the iron, that is Fe2 plus, which is responsible for formation of free radicals. So it is the main antidote for anthracycline toxicity, that is doxazoxane or ADTA. So here first it is activated by hydrolysis by the two step and it converted into diamide acid which chelate the Fe2 plus. So it is about the anthracycline derivative. Doxazoxane is also as an inhibitor of topoisomerase second but not breakage 3 strand of DNA. The additional property is important in its ability as an antidote in case of anthracycline extravasation. So this drug, antidote doxazoxane, is used when there is a leakage of anthracycline in underlying tissue. So on that condition, it is also used to detoxify the side effect of anthracycline by chelating the Fe2 plus and therefore prevent the formation of free radical. So it is about the anthracycline anti-cancer agent. Now second antibiotic which used as an anti-cancer is bleomycine. So it is an antibiotic. Bleomycine is a glycopeptide antibiotic complex isolated from Streptomyces verticillius. It is a microorganism name. Initially by Umezolua. At least 3 different fractions of bleomycine have been isolated with the clinically used product Blenoxane. So it is a Blenoxane. It is a mixture of bleomycin A2 and bleomycin B2. Of this fraction, A2 appear to be greatest important as an anti-neoplastic activity. So bleomycin is an antibiotic obtained from the microorganism. So there are 3 different fraction is isolated from this microorganism, but here the important mixture is called as Blenoxane, which is a mixture of bleomycin A2 and bleomycin B2. So it is a structure of bleomycin. So here there is a, in case of bleomycin A2, R equal to, so here is R equal to this group, then it is bleomycin A2, then bleomycin B2 having this group. So Blenoxane is a main component of bleomycin which contain bleomycin A2 and bleomycin B2, but here the bleomycin A2 is most important. Copper is found in naturally occurring material, that is in bleomycin. It contains copper, and removal of this copper is important for the material used clinically because it significantly reduces the activity. So bleomycin also contain copper, Cu, that is copper metal. So removal of this copper is important, otherwise if there is a decrease in activity. Now bleomycin bind Fe2 plus through the multiple interaction. So binding of this antibiotic to the Fe2. Again here there is a Fe2 plus ferric. So it is a binding of bleomycin to the Fe2 plus. So bleomycin bind to Fe2 plus through the multiple interaction with the amino terminal end of peptide chain. Bleomycin may itself initiate the release of iron necessary for complexation. Interaction with DNA subsequently occur through the biothiazole portion of the molecule which intercalate between GC base pair. So here if we see the structure, it contains thiazole. So it is contain the thiazole or bithiazole. So here there are 2 thiazole groups. So it is responsible for binding with DNA. So this bithiazole intercalated between GC pair, that is guanine cytosine base pair of DNA. So this interact with that DNA. Then due to this interaction under aerobic environment, Fe2 plus is oxidized to Fe3 plus in a 1 electron process with electron being transferred to the molecular oxygen, and this gives the activated form of bleomycin which has been formulated as like this. So it is a complex of bleomycin with Fe, that is called as bleomycin FeO2 complex. So after intercalation, it bind to Fe2 plus and convert into Fe3 plus. So this complex having the square bipyramidal geometry, then this result in the production of ROS, that is reactive oxygen species, in the form of superoxide and hydroxide free radical, which initiates single strand breakage of phosphodiester backbone and release of DNA bases by oxidative cleavage of 3-4- bond of the deoxyribose moiety. So here formation of this complex, that is called as bleomycin Fe2 plus, and then it inhibit the DNA. So here also there is a formation of free radical, that is reactive oxygen species, that is superoxide and hydroxide free radical, which initiates single strand breakage of DNA. So it also affects the DNA synthesis. Regarding toxicity, the toxicity profile of bleomycin is explained by its rate of inactivation, hydrolysis of N-terminal amide to carboxylic acid. So here is N-terminal. So it is here hydrolysis taken place here. So this bond is break and hydrolysis is taken place, and it form the amide to carboxylic acid, increase the pK of amino acid at carbon 2. So the pK value of this amine is increased due to this hydrolysis, resulting in a greater degree of ionization and decrease the binding to DNA. The enzyme responsible for this conversion is known as bleomycin hydrolase, which is present in most of the tissue. So the enzyme bleomycin hydrolase causes the hydrolysis at the terminal, due to which this acidity of this nitrogen is increased, pK value is increased from 7.3 to 9.4, and this causes the decrease the intercalation between the drug and with DNA. So it is the toxicity of. Now next category, that is antimitotic or Vinca alkaloid. So it is there are two drugs, vinblastine, vincristin. It is also called as antimitotic because it having the action on mitotic spindle. So it is an alkaloid extracted from leaves of Catharanthus roseus. It is a biological source of Vinca alkaloid. The alkaloids are composed of catharanthine moiety containing indole subunit and vindoline moiety containing dihydroindoline subunit joined by carbon-carbon bond. So here if we see the structure, there are two parts. This part which contain indole ring, this part, total part which contain indole ring is called as Catharanthus moiety or catharanthine moiety, and second part is called as vindoline. This one is vindoline which contain dihydroindole. So here there is a no pi bond, it is called as dihydroindole. This part is called as vindoline. So both this part attached through the carbon-carbon bond. So it is a carbon-carbon bond. So the structure consists of two parts. The part which contains indole ring is called as catharanthine moiety, and the part which contains dihydroindole it is called as vindoline, and these two parts connected through the carbon-carbon bond. Vincristin and vinblastine differ only in the group attached to dihydroindole nitrogen, which is methyl in vinblastine, and formyl group in vincristin. So if you see the difference between vinblastine and vincristin, only difference is R1. So in case of vinblastine, R1 equal to CH3. In case of vincristin, R1 equal to formyl group. This agent enters in the cell by passive diffusion. Then this agent accumulates in cell with intracellular concentration 5 to 500 folds higher than extracellular concentration. Once in the cell, the vinca bind to tubulin disrupting formation and function of mitotic spindle. So regarding mechanism of action, so these drugs first accumulate in the cell intracellularly, and the concentration of drug in cell is 500 more than the concentration of drug in extracellular fluid, where it disturbs formation of mitotic spindle. The mitotic spindle is composed of microtubule which function as a part of cell as a cytoskeleton and are important in maintaining cellular shape and also play an important role in movement of chromosome during mitosis. So as we know, in cell cycle there is a mitotic spindle which is responsible for the shape of the cell and also involved in the movement of chromosome during mitosis, that is division of cell. So this microtubule is composed of mitotic spindle is composed of microtubule protein. So in cell cycle, mitotic spindle is there which having important role in maintaining of the shape of the cell and also involved in the cell division in movement of chromosome during the mitosis. So this mitotic spindle composed of microtubule, and this microtubule is made up of alpha tubulin and beta tubulin. As a part of mitotic process, the length of microtubule will change because of polymerization and depolymerization of associated tubulin. The X-ray studies indicate that when blastine bind between the alpha and beta tubulin, and other study have shows that there are two binding site on microtubule, that is high affinity binding site which is located at the end of spindle, and there is a low affinity binding site located along the intact spindle. So microtubule having the two types of binding site: high affinity binding site which is located at the both end of the spindle, and there is a low binding site which present on intact spindle. So binding of these drug at higher binding affinity site prevent both elongation and shortening of spindle and thereby disturb its function. Similarly, binding at low affinity binding site which occur at higher drug concentration leads to breakdown of the spindle as tubulin depolarization. As a result of this action, the mitotic spindle fail to perform their function properly and chromosome do not move to the metaphase plate, or anaphase fail to occur, and it undergo apoptosis. The agent are considered specific for the M phase of the cell cycle. So in short, mechanism of action: vincristine, vinblastine disturb the function of mitotic spindle. Mitotic spindle play an important role in cell division. Mitotic spindle is composed of two protein, alpha tubulin and beta tubulin. There are two binding site, that is high affinity binding site which is located at the both end of the mitotic spindle. There is a low binding site which are located in the intact spindle. So when these drug bind to high affinity binding site, then it prevent the shortening and elongation of mitotic spindle, which is important for cell division. And if with this drug bind to low affinity binding site, then there is a breakage of this mitotic spindle. Therefore, the cell division process affected, which leads to cell death. So it is the mechanism of action of antimitotic drug or Vinca alkaloid. Regarding uses, vinblastine is a part of ABVD. It is a combination of four drugs: ABVD, A for drmajidkhan, B for Bleomycine, V for vinblastine, and D for dacarbazine. So it is a combination of four drugs: ABVD, drmajidkhan, Bleomycine, vinblastine, dacarbazine. Used in treatment of Hodgkin lymphoma. Extravasation is concerned with the Vinca, and it is treated by administration of hyaluronidase and application of heat. So when this drug is given, then there is a leakage of this drug, which is prevented by administration of hyaluronidase, that is called as tissue cement, and application of heat. The hyaluronidase hydrolyzes hyaluronic acid and a polysaccharide component of connective tissue, and this drug may increase diffusion out of area of extravasation more quickly, preventing the build-up of toxic level. So when blasting during administration, there is a chances of leakage within the tissues. Therefore, hyaluronidase is administered, which is a act as a cement. Therefore, it prevents the leakage of this Vinca alkaloid and prevent the toxicity. So it is about the antimitotic drug or Vinca alkaloid. In next video lecture, that is lecture 6, we will see about the monoclonal antibodies and the hormone which is used in cancer. So if you like the video, then subscribe the channel. Thank you very much.